Hi guys, welcome to the contraction of the skeletal muscles. So you need to be able to explain what is meant by antagonistic muscles and how they operate. We need to summarize the evidence that supports the sliding filament mechanism. We need to explain how the sliding mechanism causes muscle to relax and contract, and state where the energy is coming from. So this is our specification again. So we will be looking at the energy. We will be looking at the role of the filaments in the muscle contraction and through the whole process. So this is what we finished in our previous video. So we were looking at the differences between the sarcomere when the muscle relaxes and when contract. So this is okay, the sliding filament mechanism, and now we need to look what are the evidences for this mechanism. So what are the differences that we can look at? So we can see that actin and myosin will slide past one another when the muscle contract because of the myosin heads. So let's draw the head here. Okay, so remember there was myosin head that was pulling the filament, right? What we can see, what's the evidence for this? So sarcomere gets shorter. That's correct. That's the sarcomere when a muscle was relaxing. That's when it contracts. How is it possible? Because obviously we've got more overlaps now between myosin and actin. Z lines, so those lines here, they got shorter, as we can see. I bands get narrow. So I band was that one where we could see only actin. Okay, H zone gets narrowed. So H zone was the one where we could see only myosin. And what is the part of this sarcomere that doesn't change at all? It's the A band. So what was the A band? The A band was the length of the myosin filament. Okay, so that remains the same. So in terms of the muscle contraction, we've got obviously three stages here. So we've got the muscle stimulation. So we will be looking at the depolarization here. We've got the muscle contraction and we've got the muscle relaxation. In terms of the stimulation, there is not much difference. So let's draw the presynaptic neuron membrane. Here, imagine we've got the myofibril, so that's the muscle. So the stimulus will arrive, okay? The action potential will reach many neuromuscular junctions, okay? So those are neuromuscular junctions here. So what will happen? The calcium ion channels are going to open. So we've got calcium ion channels. They're going to open and diffuse. That's important word, through the synaptic knob. Okay. So the synaptic vesicles obviously are going to, with the acetylcholine, are going to fuse with the presynaptic membrane. They're going to now release the acetylcholine to the synaptic cleft. So acetylcholine will diffuse across the cleft and bind to the receptors on the muscular cell surface. So remember here we've got the myofibril, the receptors on that. That way the wave of depolarization has arrived to the myofibril. What happens next? Okay, don't forget about calcium ions here as well. So what happens next? Obviously the actual process of muscle contraction. Okay, so how does that happen? So this is our myosin filament with the myosin head. Myosin head it's attached to the, so ADP it's attached to the myosin head. We've got actin filament, so that thinner filament, and around the actin filament we've got tropomyosin blocking the binding site. So this is a binding site that myosin head it's aiming for. Okay, so what happens here when the calcium ions arrive? Okay, so when they are released from the sarcoplasmic reticulum, sarcoplasmic reticulum, they will cause tropomyosin molecule to change shape. That's important. So they're changing shape and they're being pulled away from those binding sites. So the binding sites now are uncovered thanks to calcium ions that arrive due to depolarization. So once they've changed the site, the myosin head now will attach to those binding sites. Remember, ADP is still there. Okay. So once they've done that, they're going to produce myosin actin cross bridges. So those cross bridges, okay, are going to be really important. Why? Because this is now the moment when the myosin head can change its angle, so it can move the actin filament along it. Once this is taking place, the ADP molecule is released. Okay, so ADP molecule was released, but what comes along is ATP. So ATP molecule will fix to myosin head, causing it now to detach. So the job was done. The head has pulled the actin filament, so now it can detach thanks to ATP. So it's going to detach. And obviously, what we want now to happen, we need to bring this myosin head to its normal position, so it will be ready for the next contraction. So what do we need for this? We need the hydrolysis of ATP, okay, to ADP by ATP synthase. So that will provide the energy for myosin head to go back to its normal position. Okay, so once they are at the normal position, okay, the myosin head reattaches, okay, to the binding site, and another process can take place. So it's like a cycle that keeps repeating. Okay, so that's the whole process of the muscle contraction. So once the muscle has contracted, obviously it's going to now aim to relax. And how does it take place? So obviously it takes place when the stimulation will decrease. Okay, so once there is no further depolarization coming along, the calcium ions now will be actively transported back to the endoplasmic reticulum, using that energy from hydrolysis of ATP. So everything comes back to normal. So what comes back to normal? So tropomyosin will now block the actin filaments binding sites again, because we won't have any more calcium ions due to no depolarization. So myosin heads will be unable to bind. Okay, so contractions will decrease and the muscle will relax. Okay, so that force from antagonistic muscle can pull actin filaments out from between myosin. So what we've got left here, we need to be looking at the energy. So we did say that obviously ATP, hydrolysis of ATP provides energy, but not only that. So because there is a high demand for energy, we would like to avoid anaerobic respiration. So to do so, we need phosphokeratin. So phosphokeratin acts as the inorganic phosphate. So this is the supply of the inorganic phosphate to regenerate ATP. So what's the main job of phosphokeratin is to store, okay, replacement using inorganic phosphates from ATP when the muscle relaxes. So here a quick activity for you. You can pause the video and spot the mistakes. Okay, so what's wrong with that answer? And the question is to describe the role of calcium ions and ATP in muscle contraction. Right, ready for the checkpoint? So what was wrong with it? Okay, in the first sentence you should say that calcium ions remove blocking molecules that uncovers binding site on actin. Calcium ions then binding to tropomyosin, and this is the old spec question. Remember, troponin, it's not the thing that you need to know for your specification. So calcium ions bind and moves tropomyosin. So that allows myosin heads to attach to actin filaments, allows sliding of the actin and myosin filaments,